So I was tasked with providing expert opinion and review of literature on the risk of pollen and compost to spread pathogens. So the pollen report is actually complete, and an overview will be presented here, but the compost project description has been written and has been put to KVH for approval, or not. So I'll just give you a revision of the biology of what we're talking about. So what does pollen actually look like? We can't actually see individual pollen grains with our naked eyes, but when we use a microscope, and so this is a scanning electron microscope, we can see that they're sort of long things with grooves along the sides. And when they're hydrated, they sort of pop out and become round. And the picture on the right is actually a light microscope. And then they germinate, and the germ tube actually comes out through those little grooves in the pollen grain. And so putting it in context, because I'm sure you're all familiar with PSA, but I'm going to be talking about some other pathogens and other groups. So I'll show you how this all fits into the overall picture. So we know that male flowers are around about 5 centimetres, or some, and the buds are a little bit less. Anthers are about 5 millimetres, and pollen is about 30 micrometers in diameter. And we can see, of course, anthers in male flowers unaided, but to see individual pollen grains, we need a light microscope. So moving down to where the pathogens are, there's pollen again at 30, but the fungal spores actually around about 15 micrometers in length of this particular fungus. But a PSA is down here at 5 microns, and the viruses are even smaller again. So this particular virus is about 20 nanometers in diameter. So we can see the bacteria, the fungi, and the pollen with the light microscope, but we can only see the viruses with the electron microscope. And putting that in context of pollen, here we have PSA actually on pollen, and you can see that they're a lot smaller than pollen itself. And then going down that step again to look at a virus on bacteria, and you can see that they're tiny compared to the bacterial cells. And putting in terms that we're all very aware about, COVID-19, the virus that causes that, is also in that size range. So talking about the outcomes of what we have done, we reviewed pollen production, kiwifruit pollen and plant pathogens that infect kiwifruit flowers, reports of pathogens on pollen of other plants, evidence for pollen transmission of bee, human and plant pathogens, mitigation methods used to control bee, human and fresh food pathogens that could be used on pollen. And a very important point when talking about pollen is that it is contaminated with plant debris. And you can see where those arrows are. Those are little pieces of kiwifruit flowers, probably filaments and other bits and pieces. We're not quite sure what they are. So whenever I'm talking about pollen, rather than say pollen and associated plant debris, I'll just say pollen, but that's what I mean, pollen and associated plant debris. And so the reason for that is part of the pollen production methods, which I'm sure a lot of you are aware of, but this is just a revision of those processes. So pollen in kiwifruit is produced on male flowers and transferred to female flowers by bees and other insects. The male flowers are harvested at the popcorn stage. So you can see the picture there of a kiwifruit bud just about to open, and that's the popcorn stage. The flowers are roughly cut, then milled by passing through a rotating drum made of a metal sieve. So that's that green thing there. And then the anthers fall through the metal sieve onto a conveyor. You can see the blue conveyor belts in that picture. The anthers are then placed on trays and dried overnight, so 25 degrees for 12 to 16 hours. And the pollen is separated from the anthers in a cyclone separator, so that's the final diagram. But flower debris with a similar mass to pollen will not be separated, and that's why the plant tissue is contaminating commercially milled pollen. So moving on to the pathogens on pollen and flowers, well, I'm going to concentrate, there actually are quite a few, but I'm going to concentrate on the ones that either are found on flowers, on pollen, or we have evidence that they are transmissible on pollen. So one bee pathogen, and we're moving down the sizes, so we're starting with the biggest pathogen, which is the fungi. So the bee pathogens, there's a disease called chalk brood, which is caused by a fungus, and it is well known and proven that this is transmissible on pollen. And when they feed bees with pollen, they always irradiate the pollen to remove all the pathogens on the pollen. This chalk brood can infect hives if you introduce contaminated pollen. So the plant pathogens, this is from other plants. We know that Botrytis cinerea is found on pollen, it infects flowers, but we don't actually know whether it's transmissible, but it is quite likely that it is. There's another fungus called Cercophora capenensis, which is pollen transmissible, and there's evidence for that. The same with Colletotrichum acutatum and Sclerotinia sclerotium. And of those pathogens, 3 are actually found on kiwifruit, but we have never investigated whether they are pollen transmitted in kiwifruit. Botrytis, you'll all be aware of, causes a storage rot. Colletotrichum acutatum is one of the ripe rot pathogens, which also rots fruit in store. And Sclerotinia infects flowers in the orchard and can cause scarring on kiwifruit. So moving to the bacteria, once more, there's a bee pathogen called American foul brood, which is a lot of evidence that it is pollen transmitted. We don't actually know whether it contaminates flowers as well, but if it's on pollen, I'd say that it probably does. There are 2 human pathogens also that contaminate pollen, and this was found during a study of people who are allergic to pollen, and they found that people weren't actually allergic to pollen itself, but to the toxins that are produced by the bacteria that were contaminating the pollen. And so, yes, those 2 bacteria are pollen transmissible. Then moving to the plant pathogens, fire blight, it's found on pollen, on flowers, and it's transmissible. Same with walnut blight, bacterial blast, which is a disease of stone fruit, PSA, which we're all familiar with. And then blossom blight, we know it's on pollen, we know it infects flowers, but we don't know whether it's transmissible. And there's 2 other bacterial pathogens, maize bacterial wilt and a disease of tobacco, Pseudomonas syringae pathophile tobacci, which are found on flowers, and we don't know whether they're pollen transmitted or not. And 2 of these bacteria are found on kiwifruit, so PSA and blossom blight. Blossom blight is also known as bud rot, so you might know it by that name. And then moving on to the Oomycetes, which aren't quite a fungus, and they can be found on flowers. So Phytophthora ramorum, which is sudden oak death, it's found on flowers. Phytophthora arecae infestans, and downy mildew of grapes. Pollen that's rotting after it lands in ponds or on the ground has actually been found to be rotted by members of this group. Chromista is the big name for the big whole group. As far as kiwifruit's concerned, Phytophthora is found on kiwifruit, but it's usually a root or a base of the vine disease. But it is possible that Phytophthora spores could get rain splashed onto flowers, but we just don't know. viruses, the smallest of all these pathogens, have only recently been discovered on kiwifruit, on an imported kiwifruit plant by Gerard Clover in 2003. Since then, there's been some work in New Zealand by Mike Pearson and his colleagues, and more have been discovered. And then the Italians have looked, and more recently the Chinese have started looking at what viruses there are on kiwifruit. So as you can see, there's more and more viruses being discovered, and there's no reason to expect that this would not carry on, so that even more viruses might be discovered the more people look for them. And of those 19 viruses, Arnaud Blouin, who works for Plant and Food Research, categorised them into 3 categories. So Ka means kiwifruit associated, so they're only found on kiwifruit. NS means non-specific, so those viruses are found on other plants as well. And then the most worrying, which is the KDV, is the kiwifruit disease-inducing viruses. And infection by those viruses will deform fruit. So of those viruses, they're all found on kiwifruit, but not all of them are in New Zealand. And there's only one which has been proven to be pollen transmitted on kiwifruit, and that's Actinidia seed-borne latent virus, and that's a very new discovery. The 2 kiwifruit disease-inducing viruses are pollen transmitted on other plants, and as are alfalfa mosaic virus and cucumber mosaic virus. And there's no viroids or phytoplasmas yet been found on kiwifruit. So mitigation methods that have been used for kiwifruit pollen include chlorine dioxide in this whole great big list. The one that worked quite well but mainly killed the pollen was acetone, so that's not a very effective treatment. And the one that worked best was actually the combination of heat, low humidity, and nitrogen. And the nitrogen was used to provide non-aerobic environment. However, none of these treatments have been commercialised as yet. And we do know that ozone can be used to treat pollen, but we're not sure what the efficacy of it is against PSA. Other mitigation methods that's been used for other plants, human medicine and fresh food processing that could be used for kiwifruit pollen include modified atmospheres, essential oils and natural products, heat shock, fungicides, biocontrol agents, or high health mother plants. So the highest risks of pollen transmission and mitigation, in my opinion, within New Zealand is PSA on orchards that don't have it, cherry leaf roll virus, and Actinidia seed-borne latent virus. Cherry leaf roll, if you remember, that causes deformation of fruit, and so that could be economically important. But the symptoms of Actinidia seed-borne latent virus are mild, as far as we know, but it's only just been discovered. From imported pollen, the highest risks are for new strains of PSA. If we import new strains of PSA that have effectors that can overcome the tolerance of Hayward and Zespri Gold to PSA, then we could be right back to where we were in November 2010. So we don't want that at all. The other high risk is Pelargonium zonate spot virus, which is another of the viruses that causes fruit deformation, and it's not currently in New Zealand. And also unknown viruses that we are not aware of as yet. To mitigate, we can use a combination of technologies. I showed you the most effective was a combination of low humidity, heat, and anaerobic conditions. We could also use high health mother plants to to control PSA. It works just as well for PSA as it does for viruses. and is used in a lot of industries when they propagate, to make sure that they don't propagate viruses as well. And this is a slide courtesy of Matt Dick to show what is actually being done in the pathway management plan to ensure safe movement of pollen. So KVH realises that growers have a lot of compliance to have to deal with, and so the flower suppliers already operate to a biosecurity plan. So one of the specifications is to continue operating to the biosecurity plan, and if there's any specific requirements for high-risk organisms. So obviously for if you're supplying pollen to PSA non-detected orchards, then it's a good idea to source the flowers from PSA non-detected orchards. And with the mills, they need to register with KVH and to make sure that there's good record keeping so that traceability is possible in the event of something going wrong. Good hygiene practices, and source flowers from compliant suppliers. And the suppliers, registration and traceability. So it's a bit like a COVID-19 system where good records are kept, traceability is possible. So in the event of something happening, things can be traced back to the source and dealt with as soon as possible. But one thing that has come out of the pollen review is that it has confirmed that the existing protocols are adequate for managing the risk. So with the compost, a project description has been prepared, and compost actually is a variety of things, and for the proposed review, we're going to be dealing with raw compost. So raw compost includes fish and cow manure, chicken manure, and your organic matter from gardens or from parks. But it also includes vermicompost and vermicast, which from worms, and mulch. So if it's approved, the report is due in late February 2021. So finally, thank you for your attention. And thank you to KVH and Zespri for funding, and Sonia and Matt and Erin for their comments as well. So are there any questions? Yeah.
Kerry, your use of the term pollen in the presentation, does that specifically exclude bee-transferred pollen?
Not specifically, no. We know that commercially milled pollen is contaminated with plant tissue. Bees would also be contaminated with plant tissue, just the way that they deal with the plants. I doubt if they wouldn't be transferring bits and pieces of plants as well as pollen.